Where do you come from is one of the most frequently asked questions when people first meet. Regardless of the social unit in question, country, state, city, family, origin stories tend to be of interest to most of us as it helps place our perceptions of people into context. Now we can apply the same learning to studies of peptides. Identifying distant related peptides provides clues as to the common origin and informs us about the properties that underlie their current structures and function. Information about the evolutionary history of peptides is particularly interesting when it comes to venom peptides, often referred to just as toxins, which are found in spiders such as this one here. Toxins generally have high potency and selectivity for particular receptor and ion channel subtypes, which has made them useful pharmacological tools and attractive leads for development of peptide-based therapeutics and bioinsecticides. Revealing the evolutionary connections between toxins and their ancestral non-toxin precursors therefore provides insight into the molecular traits that are important for attaining the required levels of potency, selectivity, solubility, and stability that often characterize these toxins. However, studying the evolution of toxins is not always an easy task, and in a sense, the traits that make toxins particularly interesting from a biological perspective are the same traits that make their evolutionary history challenging to study. In many animal venoms, such as spiders, most toxins are cysteine-rich peptides. These are stabilized by several internal covalent disulfide bonds, which provide them with great stability that is characteristic of toxins. The disulfide bonds are also so important for the structure of the peptide that most other residues can be mutated without perturbing the overall structure of the peptide. Toxins are thought to originate from housekeeping proteins that are recruited into the venom, where they acquire new functions through evolution by positive selection. During this process, almost every amino acid can be mutated, leaving very little information for investigating the evolutionary history. We believe 3D structures are largely overlooked evolutionary tools that provide a way around many of these problems. Because although the sequences of two related toxins may be almost completely different, their overall structures tend to be very well conserved. 3D structures can therefore help us identify distantly related peptides that allow us to come up with more informed hypotheses regarding the deep phylogenies of toxins and other cysteine-rich peptides. 3D structures also help place mutations and sequence variability into biological context, thereby identifying the structural adaptations that underlie their unique properties. Currently, however, phylogenetic trees tend to be used to infer protein structures rather than the other way around. Structural biology methods have evolved to the point where high-resolution peptide structures are attainable within a matter of weeks rather than months, and at a reasonable cost. This is thanks to advancements in technology such as nuclear magnetic resonance, or NMR, like this facility behind me here. At the same time, improvements in genomic and proteomic platforms have caused an increase in the rate of deposition of novel peptide sequences into public databases. This has led to an increase in both accessible sequence data and the ability to generate 3D structures. There is therefore now a need to develop methods for combining these datasets so we can unravel the deep evolutionary histories of proteins and peptide folds. With more information and by using such an integrative approach, we hope to gain meaningful insight into the evolution of cysteine-rich peptide folds and find out where they have truly come from.